Welcome to The Explainer. Today, we are diving deep into the molecular mystery at the very heart of the neurodegenerative disease ALS. And it's a story that really hinges on one single character inside our nerve cells.
Okay, let's just start with the single most important number in modern ALS research, 97%. I want you to remember that number, because it's not just some statistic. It is a massive, massive clue that has completely changed how scientists think about this disease. So what is it? What's the one thing that unites almost every single person with ALS, whether their case is genetic or it just seems to appear out of nowhere? Well, the answer isn't what you might think. It's not about lifestyle or environment. It's about one single protein going rogue inside the nerve cells. And that answer? Well, it lies with a protein called TDP-43. Understanding its story, how it's supposed to work, and how it goes so wrong is absolutely fundamental to understanding ALS today. Now, TDP-43 isn't new. It was actually identified way back in the 1990s. It's this crucial manager that you can find in pretty much every cell in your body. But, as we're about to see, its job inside our very long, very complex nerve cells is uniquely important, and it turns out uniquely vulnerable. Okay, so let's dive into what this protein is supposed to be doing. Because in a perfectly healthy neuron, TDP-43 is the unsung hero of our story, quietly making sure everything is running just right. The best way to think of TDP-43 is like a master librarian. It works inside the cell's command center, which is called the nucleus, and it meticulously manages the flow of all the genetic instructions, the RNA, making sure the right messages get processed, get stabilized, and get sent out at exactly the right time. So, the bottom line is this. This one protein ensures the cell's entire machinery runs like a well-oiled machine. And this is especially critical for our motor neurons. I mean, these are cells that have to maintain connections that can be over 3 feet long. Without that kind of precise management, they just can't survive. But then, in 97% of ALS cases, something goes terribly, terribly wrong. Our hero abandons its post. It walks out of the library, and in doing so, it becomes the villain of the whole story.
And the core problem, it's shockingly simple. This is the single most important event in ALS pathology. The protein physically moves. It moves from where it belongs, the nucleus, to where it absolutely does not belong, the outer part of the cell, the cytoplasm. And this one small journey has catastrophic consequences. You see, this move creates what scientists call a devastating double hit on the neuron. First, the cell loses its essential manager inside the nucleus. That means chaos for its genetic instructions. And second, the cell now has to deal with a new toxic presence building up outside in the cytoplasm. The cell is literally being attacked from two sides at once. So let's follow the chaos, shall we? Let's see how this one change, this one protein getting out of place, sends these ripples of destruction through the entire neuron, basically grinding all of its essential functions to a halt. You know, without its manager, the cell just starts making silly mistakes. One of the clearest examples is the appearance of something called cryptic exons. You can think of these as hidden, faulty instructions in the genetic code. TDP-43 is normally there to say, nope, we skipped that part. But with TDP-43 gone, those faulty instructions get included, leading to the production of totally useless or even harmful proteins. And at the exact same time that rogue TDP-43 in the cytoplasm starts forming these toxic clumps, or aggregates. These clumps physically clog up the works. They overwhelm the cell's garbage disposal systems, they damage the cell's power plants, and they totally disrupt the transport highways that are supposed to keep the neuron alive. It's really just a slow, systematic shutdown from the inside out. This whole unified story only really became clear in 2006. That was a huge, landmark year. It's when researchers finally figured out what those mysterious protein clumps they kept seeing in ALS neurons were. And the culprit? You guessed it, was TDP-43. And, crucially, they found the exact same protein causing trouble in another brain disorder, FTD. And that discovery, wow, it just blew the doors off the old way of thinking. ALS isn't some isolated disease of the motor system. No, it's part of a much larger family, a spectrum of related conditions that are all linked by the very same core problem. Think about it like this. You know how we always associate Alzheimer's with the tau protein? Or Parkinson's with a protein called alpha-synuclein?
Well, we now know that the vast majority of ALS cases and a lot of FTD cases are fundamentally TDP-43 diseases. They all belong to this family of disorders where a specific protein just starts to misbehave. So the symptoms a person gets simply depend on where in the nervous system TDP-43 is causing the most trouble. If it's mainly hitting the motor neurons, you see the muscle weakness of ALS. But if it's hitting the frontal and temporal lobes of the brain, you see the cognitive and behavioral changes of FTD. It's the same villain, just a different crime scene. But here is the most important takeaway from all of this. This is the real source of hope. Because scientists have identified this common enemy, this villain, in 97% of cases, they finally have a clear, unified target for developing therapies.
And that has created a real roadmap for developing drugs. Researchers are now exploring so many exciting strategies, and they're all aimed at fixing the core problems we've just talked about. Things like trying to force the protein back into the nucleus, or stopping it from clumping up, or even just helping the cell clean up the mess.
Of course, it's not that simple. The goal isn't to just get rid of TDP-43 altogether, because, remember, it's an essential hero in our healthy cells. The real challenge, the puzzle, is to figure out how to gently guide this hero-turned-villain back to its proper job, to restore that delicate balance without causing even more harm. So, while there is still so many challenges ahead, the discovery of TDP-43's central role has given us a powerful, unified path forward. It has focused the entire research field on one single fundamental question. If we can figure out how to fix this one protein, could we finally stop this devastating disease?